research field as well as the clinical practical management in lung cancer management. Following the talks, we also have very interesting case-based presentations by our Year 5 medical students. Now, before we kickstart the program, perhaps allow me to go through some of the housekeeping announcements for today's event. Can I have the housekeeping slide, please? Thank you. So please be known that the microphones will be muted throughout the entire webinars. And you do have any questions, please type it in the chat box, and the questions will be moderated later during the Q&A sessions. The webinar will be recorded, and the video recording will be posted on the websites of the on the Center for Cancer Research as well as the Center for Stem Cell Research. The last rules is very important. Please rename your name in your to your full name, your Zoom ID to your full name to facilitate the preparations of the certificate of the attendance as well as the collections of CBT or soft skill points. Next, I would like to introduce or invite Dr. Teo, who is the organizing chairperson of this webinar, to deliver her opening address. Dr. Teo.
Right, thank you, Dr. Lee. Can you hear me?
Yes, very clear.
All right. So a very good morning to all our distinguished speakers, organizing committee members, and our participants. All right. First of all, thank you to everyone for taking the time out on a Saturday to attend this webinar. Right. The International Webinar Series is an annual event organized by the Center for Cancer Research and the Center for Stem Cell Research. Right. Through the webinar series, we aim to bring together medical and research experts from different specializations to speak about the different types of cancer and the latest advancement in research and clinical practice in cancer management. For our first webinar of this year, we are happy to bring to you a lung cancer webinar, and we have invited experts in this area to share their knowledge in the research development and clinical management of lung cancer. Our first two speakers will talk about the oncogenic role of circular RNAs in non-small cell lung cancer and also lung cancer stem cells. And our third and fourth speakers will provide updates on the clinical management and pulmonary surgery in lung cancer. We've also put together by our Year 5 MBBS students a case presentation on non-small cell invasive adenocarcinoma of the lung. And this will be followed by a discussion of the case by our panel of experts. So through the talks and the case presentation, we hope to provide a virtual platform for learning and discussion to generate new ideas and establish new collaboration among the participants of this webinar. Finally, I wish everyone a successful and fruitful session. My sincere appreciation to all the speakers for their support and also to the organizing committee members for their hard work to make this webinar a successful one. Thank you, everyone. Back to you, Dr. Lee.
Thank you, Dr. Teo. So please allow me to introduce Dr. Wang, the Research Assistant Fellow in the Department of the Medical Research, Taipei Veterinary General Hospital, Taiwan. She got her PhD from the Department of Biochemistry and Molecular Biology from the University College London, UK, supervised by Dr. Ivan. Currently, she is working on the mechanism involving the drug resistance and tumor metastasis, as well as the effect of microenvironment on these tumor properties. So let's welcome Dr. Wang Monlian.
Thank you for the introduction. Let me just share my slide. Okay. Thanks for the introduction, and it's my honor to be invited to the conference. Today, I'm going to share my recent results in the investigation of lung cancer-related circular RNA and its molecular involvements in cancer progression. Lung cancer is the leading cause of cancer-related death and responsible for highest new cancer cases throughout the world. With around 2.1 million new cases and 1.8 million cancer-related deaths in 2018. The predominant type of lung cancer is non-small cell lung cancer, accounting for 85 to 90% of lung cancer cases. Non-small cell lung cancer can be further divided into several subtypes, including squamous cell carcinoma and lung adenocarcinoma. In Taiwan, lung adenocarcinoma accounts for nearly 70% of non-small cell lung cancer, or in other words, it accounts for more than half of all lung cancer cases. The EGFR mutation is considered as one of the most important oncogenes to cause lung cancer mutations and tumor progressions. Around 35% of Asian and 10% of Caucasian patients have mutations on EGFR genes. Because lung cancer is not easy to detect at the early stage, only less than 30% of diagnosis cases are operatable. Nevertheless, despite the current development of EGFR target therapy and immune blockade therapy, the treatment outcome is still very poor. A considerable portion of patients develop resistance to these current therapies. Therefore, we want to ask a question. Can we identify an easy accessible biomarker that reflects the disease status of lung adenocarcinoma patients and their response to clinical therapy? Such as a biomarker that can be detected in patients' blood would be an ideal marker. And therefore we place our focus on circular RNA. Circular RNA is a type of non-coding RNA coming from back splicing of pre-mRNA processing. It was first discovered in 1990s in yeast. Until now, the function and majority of the circular RNA is still unknown. Like other non-coding RNAs, circular RNA was considered as non-functional byproducts of the mRNA splicing processing. Only till the recent decades does researchers start to discover the hidden molecular involvement of the circular RNA in cellular functions. Some circular RNAs has been shown to code for proteins, and some circular RNA have recently shown have the potential as gene regulators. They could regulate genes through like an mRNA sponge, which is the most well-studied function of circular RNA, or the protein sponge to bind some like RNA binding protein, or involves in transcription or translation regulation. However, the biological function of most circular RNA are still largely unclear. And it is only recent that people start to realize that circular RNA may involve in several disease pathogenesis, including cancers. The figure in the left-hand side lists out a few example circular RNAs that are implicated in cancer and the cellular functions they were reported to involve. In cancers, circular RNA could be both either oncogenic or tumor suppressive, depends on the mechanism and targets it acts on. So to identify the lung cancer-related circular RNA, we firstly use 3 cell lines for circular RNA sequencing. The BS2B is the immortalized bronchial epithelial cell line, representing a normal lung cell. The A549 cell is a lung adenocarcinoma cell line with wild-type EGFR. And the HCC827 is also in a lung adenocarcinoma cell line, but with active mutation EGFR. We try to find the circular RNA with an increasing expression level along with the activation status of EGFR. And with a pattern and with a significant p-value, we identify 9 potential circular RNA targets. And after a careful validation by qPCR, we narrowed down our target to C190 and 1649. We did several validations to assure that transcripts were measured. The transcript we measured were the circular RNA rather than their linear counterparts. This includes single sequencing of the specific splice junction of the circular RNA, and to confirm that the transcript we detected occurred only in the cDNA sample, but not in the genomic DNA samples. Because our aim is to identify circulating circular RNA, so we need to know that if these identified circular RNAs can be secreted or not. And due to the very little amount of secreted RNA, either in culture medium or in patient's blood sample, we developed a digital droplet PCR, or in brief, ddPCR, detection protocol, which is believed to be 1,000 times more sensitive than traditional qPCR. This is a test of GAPDH copy numbers with a zero dilution of the cell samples to show the linear regression of the ddPCR detecting method. And using this method, we did able to detect C190 and 1649 in the condition medium from several lung cancer cells. We then recruit a cohort of 231 lung cancer patients and 41 healthy donors to analyze the plasma levels of the two circular RNAs and correlate the levels with the disease status. First of all, we showed that in the patient sample, we did able to detect the C190 and 1649 circular RNA in patient's plasma. And both circular RNA were expressed higher in lung cancer patient in comparison to the healthy donor. And patients with stage 4 demonstrate significantly higher expression level of C190 comparing to the patient with stage 1 and 3. And a similar result was also obtained when stage 3 and 4 were compared with the stage 1 and 2. Circular RNA 1649 also showed the same trend, but the difference was not reached the statistical significance. And then the primary tumor size was positively correlated with the expression level of the circular RNA in patient's plasma. We then further explored the potential of the two circular RNA to predict and monitor disease progression of advanced lung cancer patients undergoing immunotherapy. First of all, we demonstrate that the level of C190 in plasma was related to exothoracic metastasis and distal metastasis. So patients with 3 to 4 metastatic organs has a higher expression level of circular RNA 190 in comparison with those with 1 to 2 metastatic organs. And the same phenomenon was seen in patients with extrathoracic metastasis. And moreover, patients with partial regression after the immunotreatment express a lower level of C190 compared with patients with stable disease or progressive disease. And then we use this cohort of patients, we use a bigger cohort of patients to analyze the correlation between the expression level of the circular RNA and the overall survival. We see that patients expressing high level of C190 demonstrated a significantly worse overall survival but those with a high level of circular RNA 1649 didn't show a significant difference between the two groups. So collectively, our data suggests that the two circular RNA, especially the C190, can have the potential to be used as a marker to monitor the patient's disease status. We then performed a serial blood test from lung cancer patient for evaluating the association between treatment response and a serial change in the level of the two circular RNAs during the treatment period. The change of the plasma circular RNA levels during immunotherapeutic treatment were monitored in totally 6 lung cancer patients. The three of the patients on the top panel who received anti-PD-L1 treatment had progressive disease and also demonstrated an obviously increasing level of circular RNA 190 and 1649. On the other hand, the 3 patients at the bottom panel shows decreased tumor size following the treatment and were also accompanied by an obviously decreased C190 expression level, despite of their PD-L1 expression level. So overall, these data suggest that the two circular RNA, C190 and 1649, may be utilized as novel biomarkers for the effectiveness of immunotherapy, regardless of the patient's PD-L1 expression level. Following the identification of the prognostic value of C190, we want to ask, does C190 involved in the disease pathogenesis signaling in lung adenocarcinoma? In fact, C190 not only elevated in cancer patients' plasma sample, but also elevated in lung adenocarcinoma tissue sample in comparison to the adjacent normal counterparts. And this phenomenon was not observed with its parental gene, the CNIH4 gene. This is the gene where the circular RNA C190 comes from. In our cell model, we found that the cellular level of C190 can be enhanced by activating EGFR signaling pathway with EGF treatment. And along with the increasing expression of C190 induced by the EGF treatment, in our model, 2 of the EGFR downstream pathway, the AKT and the ERK pathway, but not the STAT3 pathway, were activated alongside with the increase of C190. So then we apply 2 inhibitor to the cell. The LY is the AKT inhibitor, while the PD is the ERK pathway inhibitor. And after adding the inhibitor to the cell, we observe that only PD inhibitor can successfully suppress the EGF-induced C190 expression, indicating that C190 is downstream of the EGFR and ERK pathway in lung cancer cells. We then constructed overexpression plasmid of C190 and overexpress it in A549 lung adenocarcinoma cell. We found that ectopically overexpression of C190 in the A549 lung cancer cell can also induce the phosphorylation of ERK and not EGFR. So that means the C190 and ERK signaling form a positive feedback regulatory loop downstream of EGFR. In vitro functional assay analysis show that overexpression of C190 in both A549 and H12799 lung cancer cell line increases cell proliferation, as well as the cell migration and cell invasion ability of lung adenocarcinoma cell lines. In line with our in vitro data, our xenograft tumor model demonstrated enhanced tumor growth of the C190 overexpressing cell in the mice. And the expression of C190 and the enhanced cell proliferation in the xenograft tumor sample were later confirmed by FISH assay and the Ki67 staining. Now we know that C190 is an oncogenic circular RNA. We want to bring down its expression to see if this could be a way to suppress tumor growth of lung adenocarcinoma. But how can we target C190 without touching its parental gene? So then we apply and introduce the CRISPR-Cas13 RNA targeting system to this research project. The CRISPR-13 is a single effector RNA-guided RNase. Naturally, it is a system to destroy viral single-strain RNA, but now it has been developed and broadly used to knock down RNA, perform RNA modification, and even RNA imaging in live cells. So we apply this technique to knock down C190 in A549 and HCC827 cell lines. This is the knockdown efficiency. And then, in line with our previous observation, knockdown of C190 suppressed the ERK phosphorylation in both cell lines, but did not change the phosphorylation status of EGFR. And further, it also inhibit the cell proliferation in A549 and HCC827 cells, and also suppress the clonogenic growth of the lung cancer cell, as well as reduce the cell migration ability. So we then want to prove that in mouse model that targeting C190 can suppress tumor growth in vivo. We inject A549 cell with stably overexpressed Cas13 protein, and then 2 weeks later, we inject the guiding RNA targeting the C190 on the tumor site twice a week, and then monitor the tumor growth up to 1 month. Our xenograft tumor model showed that injection of the C190 targeting guiding RNAs can suppress the growth of subcutaneous tumors, as well as reduce the Ki67 activity of the tumors. And the CRISPR-Cas13 mediated knockdown of C190 was also confirmed by FISH assay. So in conclusion, our data showed that the C190 is an oncogenic circular RNA that is downstream of EGFR and enhanced tumor progression by activating MAP kinase pathway. And that knocking down of C190 reduced cell proliferation, migration, and tumor growth in vivo. This study provides evidence of the prospect of using CRISPR-Cas13 RNA editing system to target circular RNA for therapeutic purpose. So this is the end of my presentation, and my sincere acknowledgement goes to all the members in the molecular oncology lab in the Department of Medical Research in Taipei Veterans General Hospital. I would particularly thanks to Dr. Afiz Ishola, who put lots of effort in this project and just graduated from this lab early this year. And also Dr. Shuhua Cho, the director of the Department of Medical Research, who gave us a lot of support on this project. And finally, thank you for all of your attention to my presentation. Thank you.
Thank you, Dr. Wang, for sharing your research work with all of us. It's indeed a very interesting topic. For those of you who have burning questions, please just type in the chat box first. Yeah, we will moderate the questions later during the Q&A sessions. Thank you again, Dr. Wang. So next, we will move on to our next speaker, who is I have the privilege to introduce to you all Dr. Narashikin Zakaria. Dr. Narashikin actually has obtained her PhD in cancer genetics from University of Science Malaysia in the year of 2017. Following that, she she was a postdoctoral research fellow in the sorry about that. She was the postdoctoral research fellow in Advanced Medical and Dental Institute, University of Science Malaysia, Penang. During that time, she had experiments to analyze the role of mesenchymal stem cells in cancer development. During that time, she has supervised and trained postgraduate students, both locally as well as internationally. She also has published in a lot of index journals, conferences, proceedings, as well as chapters in books. So today we are honored to have Dr. Narashikin to speak to us on the, I believe it's her research work as well, on targeting lung cancer stem cells in research and clinical settings. Dr. Narashikin?
Thank you for the very kind introduction. Okay, so I will share my slides. Move on. Okay, good morning, everyone. Okay, you guys can see my slides. Okay. Today I will talk about targeting. My title for today is about targeting lung cancer stem cell in research and clinical setting. So this is my. I've been involved in research on cancer stem cell for more than 7 years. So this topic is one of my topic that I've published in review article in 2017. Okay, so I will share with all of you about this topic today. Okay, before we start, I think I need to close my video because the internet connection here is a bit slow. Wait. Okay, so before we start, I would like to ask these questions. Will we win the war against cancer? So as for me, finding cure for cancer is like a constant race with new findings keep appear, especially with the computational advancement that we have nowadays. So for example, like 20 years ago, scientists didn't know that cancer might develop from cancer stem cell, not until 20 years ago, where they find that cancer stem cell might be the cause of the cancer. And until now, a new information keep coming from research and give us more information about cancer stem cell. Okay, so still until now, we still face a big challenge in lung cancer. So until now, lung cancer is still remain as the leading cause of cancer-related death worldwide. Okay, so the main problem is that the cancer is always detected at late stage, where when the patient came, they already has a very advanced stage cancer. Okay, so this making them very difficult to survive. Not until 5 years when they receive the first treatment. Okay, so the treatment for lung cancer remain challenging. So due to one of the reason is drug resistance, and also because of the presence of metastasis and also the relapse. So we already know that scientists have found that the cancer stem cell might be the reason behind all this problem. Okay. So we now will explain to you what is cancer stem cell, okay, and we will see how and why this cancer is responsible for the failure of cancer treatment. Okay, so by definition, cancer stem cell can be defined as a cell within tumor that have a self-renewal capacity, and this cell can grow and cause heterogeneity within tumor cells. So cancer stem cell can only be defined experimentally if this meet these two criteria, self-renewal and give rise to heterogeneous cells. So apart from cancer stem cells, other terms such as tumor-initiating cells, tumorigenic cells, and tumorigenic cells are also used to describe the putative cancer stem cell in tumor. So what are the criteria of this cancer stem cell? So based on the name, we know that cancer stem cell is a cell that shares similar characteristic with normal stem cell. So for example, this cell possesses a stemless characteristic. They can self-renew, they can undergo asymmetric division to give rise to heterogeneous cell populations. But these cells are very tumorigenic, meaning that they can give rise to tumor or they can initiate tumor, and these cells are immortal and they also express a high level of multi-drug resistant protein which responsible for cancer resistant, and they are also highly metastatic. Okay, here I want to show with all of you a very interesting image of cancer stem cell being isolated in lung cancer mouse model. Okay, so then they try to test the cell by isolating the cell, the SCAR1 ABCG21, in sample of lung cancer patients, and then they injected the isolated cells into mice, and they see that most of the cells are localized into the small bronchial area, and then the tumor that form from the cell resemble the original tumor in the cancer patient. Okay. So this result shows the presence of cancer stem cells. Okay, next I will show to you the history behind the cancer stem cell discovery. So scientists have observed the presence of cells that behave like stem cells from years ago, but until then, they do not know yet what is the cell until the late 90s, where a group of scientists led by John Dick, they managed to isolate the first cancer stem cell in acute myeloid leukemia. Okay, so since the initial study, there have been an intense research on cancer stem cell where scientists has tried to isolate cancer stem cell from various types of tumor cell using a similar approach used to isolate normal stem cell. And as for lung cancer, the first report on lung cancer isolation was back in 2018. Okay, so the study was done by a group of scientists led by Ramo, where they identify cancer stem cell population by using a CD133 as a surface marker, and then they isolate the cells in small cell lung carcinoma and also non-small cell lung carcinoma patient samples. So when they isolate the cell, they test the cell for the ability of the cell to grow into tumor sphere, which indicate their self-renewal characteristic. And then they transplanted the cell into nude mice to see whether the cells have the ability to initiate tumor formation in nude mice. And then they also shows that the cell that they isolated, the CD133 positive cells, are more resistant to various types of chemotherapeutic drugs as compared to CD133 negative cell. So here they prove that the CD133 marker can be used to isolate and identify cancer stem cell in lung tumor. Okay. This slide shows the common method that being used in the lab to isolate cancer stem cell. So basically, cancer stem cell can be isolated by labeling the cells, the tumor cells, with specific biomarker. Okay, so the biomarker that being used is including the CD133, CD44, CD90, and many more. Okay, so since then, scientists has tested all these markers and see whether this marker can isolate cancer stem cell. Okay, so then the cancer stem cell can be sorted out by using fluorescent activated cell sorting. cell sorter or by using magnetic sorting. Okay, so after that, when we scientists will get two different population of cancer stem cell and non-cancer stem cell, and then they can use these two separate cells for the downstream testing. Even though studies have shown that single marker can be used to isolate cancer stem cell, however, there are some opinion that that saying that single marker is not enough to isolate cancer stem cell, and they call for the use of multiple marker combination, or they suggest to combine biomarker with other functional assays. Okay, this is our own study that we did, where we use a double marker, which is the CD44 and CD166, and also we also use the triple marker to select for the more stringent cancer stem cell from lung cancer cell line. Okay, so from this study, we found out that by combining 2 or 3 markers, we are able to isolate a more stringent cancer stem cell population. So this is some of the functional assays that are being used in the lab to identify and to characterize cancer stem cell in lung cancer. So for example, these subpopulation assays and ALDH assays has been used to isolate cancer stem cell based on the facts that cancer stem cell express high level of multi-drug resistant genes such as ATP binding cassette, ABC, and also ALDH genes. Apart from these subpopulation assays and ALDH assays, there are also tumorigenic assays where we use this assay to test for the tumorigenic capacity of the cancer stem cell. So the theory is that when we isolate the cancer stem cell and we transplant them into the nude mice, these cells has the ability to form a cancer. Okay, so the number that needed to form the cancer is very low as like 300 number of cells. Okay. So the next assay is the chemo-resistant and hypoxia-resistant assay. So these assays are based on the ability of cancer stem cells to survive chemotherapeutic drug, and the cell can flourish in hypoxic condition. So these assays are commonly used to enrich the cancer stem cell frequency in tumor sample. So the next assay is the 3D culture. So the most common one is the sphere culture. Okay, so these assays are used to test the ability of cancer stem cells to self-renew and undergo asymmetric division. Okay, so they are also commonly used to enrich cancer stem cell frequency and used to maintain cancer stem cell population for a longer time. Okay, so what I want to highlight here is the newest addition to the 3D culture, which is the organoid culture. So organoid culture is a latest advancement in cancer research. Okay, this 3D culture is based on the ability of stem cell to give rise to multiple cell types. And then the cell that form can self-organize and resemble the original organ. And for the case of cancer, they can form the original cancer similar to the original tumor in the patient. So this model is a very good model to test for any drugs, screening for drugs, since they give a better representation of human organ and human complexity as compared to the animal model. Okay, now we know that cancer stem cell is the main culprit in cancer. So if we eliminate cancer stem cells, or logically speaking, we can cure cancer. So how do we kill cancer stem cell? Okay, since we know that cancer stem cell is resistant to chemo drug, so we need a new agents or new treatment that can specifically target and eliminate stem cell. So among the approach that can be used is by targeting the cancer stem cell associated biomarkers, and we can also targeting the transcription factors such as SOX2, OCT4, KLF4, and CMYX. And then we can also target this cell by blocking the signaling pathway that maintain the stem cell that maintain the cancer stem cell, and also we can target the microenvironment. And lastly, we can try to develop an immune therapy that can target these cells. So this is one of the example of research showing that by downregulating one of the stem cell transcription factors in cancer stem cell could suppress the growth and metastasis of lung cancer. So as we can see here, the SOX2, which is the stem cell transcription factors, was knocked down using siRNA. And then after knockdown, we can see that the site population number, so the SP cells here is the cancer stem cell in the lung samples. So the SP cell number was reduced and the number of cells undergo apoptosis was increased. Okay. Then when they transplanted the cancer stem cell into nude mice, they observed that the cell do not form a tumor, meaning that the tumorigenicity potential of the SOX2 knockdown cell was reduced significantly as compared to the cell that did not undergo the SOX2 knockdown. Okay, so this experiment indicate that cancer stem cell had lost its stemness potential due to the reduction of SOX2 expression. Okay, next approach is by targeting the cancer stem cell associated signaling pathway. Okay, these are some of the important pathway in cancer stem cell, such as Notch pathway, Hedgehog pathway, WNT pathway, and NFKB pathway. All these pathway play a very important role in maintaining cancer stem cell stemness and proliferation. Okay. So I list down here some of the some of the agent that has been developed to specifically target this particular pathway. Okay. So here is our own study where we block the activation of NFKB signaling pathway, and from the study we managed to show that by inhibiting the activation of NFKB signaling pathway using small molecule inhibitor, we managed to reduce the stemness characteristic of lung cancer stem cell. So from the study, we managed to show that by targeting the signaling pathway could be one of the approach to target cancer stem cell. Then we can also target cancer stem cell by targeting the cancer stem cell microenvironment. Okay. So studies has proven that cancer stem cell microenvironment is very crucial in supporting cancer stem cell growth. So cancer stem cell microenvironment is comprised of multiple types of cells, such as endothelial cells, cancer-associated fibroblasts, cancer-associated mesenchymal stem cell, and macrophages. So all these cells will secrete some factors that play a role in supporting the growth of cancer stem cell. So by inhibiting all these signals, we might be able to suppress the growth of cancer stem cell. Okay, the last approach is by using immune-based approach to target cancer stem cell. Okay, this figure summarizes some of the approach that can be used to target this cell. So what I want to highlight here is the PD-L1 inhibitor and also the CAR-T cell therapy by using CAR-T and NK cell therapy. So these two approach is considered as the new approach used to target cancer cell, and also can be used to target cancer stem cell and also its surrounding microenvironment. So here I list out some of the agents that being developed to target lung cancer stem cell, and all of these agents are now already entering the clinical trial. Okay, so for example here, most of the drugs are now in the phase 2 of the clinical trial, and the PD-L1 blocker for non-small cell lung cancer are already entering the phase 3 of the clinical trial. So we hope that all of these drugs can give a promising results in treating cancer stem cell. Okay. So now we came to the conclusion. Okay. So despite many controversies surrounding cancer stem cells, still potential drugs, vaccines, antibodies, and immune therapy has been developed to target cancer stem cell. And now all the drugs are start to enter the clinical trial. So we hope that all this development may open up a possibility for more effective cancer treatment. So, however, there are several issues need to be addressed to effectively eliminate cancer stem cells. So, for example, the characteristic of many cancer stem cells are not well identified. So, we need more research to better understand these particular cells. And also, the use of immune deficient mice do not really represent the biological complexity of the tumor cell in human. So, we hope that the use of better models such as the organoid model, which give a better representation of biological complexity, can be used to replace the animal model. Okay, and finally, the cancer stem cell exists in a specific niche or in the microenvironment. So we need to study more on the function of or the role of microenvironment in supporting cancer stem cells. Okay, so with that, I will end my presentation. Okay, so thank you.
Thank you, Dr. Nora Shikin. Thanks for the very informative presentation. Okay, now I would like to invite our third speaker, Dr. Ivan. He is the clinical oncologist. He obtained his fellowship of the Royal College of Radiology in clinical oncology from UK in year 2013. And in year 2012, he obtained his Master of Business Administration from UTAR. Currently, Dr. Ivan is the consultant for the clinical oncology in Sunway Medical Center and Sunway Medical Center Velocity. At the same time, he also the consultant of the clinical oncologist in Tung Sin Hospital. So without further ado, we are welcoming Dr. Ivan. Thank you.
Excuse me, Dr. Yan, there is no audio here. Huh?
Dr. Yan, still no audio.
Is still no audio?
Yes.
Hold on.
Thank you, chairpersons, and thank you for the kind introductions. Good morning to
Okay,
everyone.
now Dr. Yen.
Dr. Ivan Shu, and today I'm given the topic of management of lung cancer and our focus in the immunotherapy treatments. Okay, I'm Dr. Ivan Shu, the clinical oncologist from Sunway Medical Center. And first of all, I will talk about the burden of disease. Then I will talk about the systemic treatment for stage 4 lung cancer, particularly immunotherapy and the others. And immunotherapy, the past and present, immunotherapy side effect, and I will share some cases. So first of all, we look at the Malaysian National Cancer Registry. It's a report 2012 to 2016, it's the latest one. And we can see a lung cancer ranked number 3. It's all the lung cancer in Malaysia, male and female. If you divide them into male and female, colorectal rank number 1 in male, and lung cancer will be the second one. For female, lung cancer rank number 4. So it's not an uncommon cancer in Malaysia. So each year we have about 2,000 cases diagnosed. Male is a bit more, 1,004. Female is about 600 to 700. So it's about 2,000 new cases per year. So like all other kinds of cancer, the cancer means the lung cancer diagnosed after about 50 years old, the increase and the risk of getting lung cancer will be more with the advance of age, as it peak about 60 to 70 years old. Most of the cancer patients presented with stage 4 lung cancer, about 80% of them. And very rarely we get stage 1 to stage 2 cancer in Malaysia. So it's a late presentation is one of the major problems. So let's just look at the stage 4 lung cancer treatment modality. We used to treat stage 4 lung cancer with chemotherapy. The effect is not good at all. So, but we have more and more these kind of new forms of systemic therapy like immunotherapy and targeted therapy and also radiations. Let's focus in these targeted therapy and immunotherapy. So for non-small cell lung cancer, and this is NCCN guideline, US guideline they use commonly by oncologists in Malaysia. For advanced and metastatic disease, non-small cell lung cancer, we divide them into the squamous cell carcinoma as well as a non-squamous cell carcinoma. So very commonly in non-squamous cell carcinoma, we can find some targetable mutations. For those without targetable mutations, so we'll do a PD-L1 testing. So this is the marker for immunotherapy. For squamous cell carcinoma, it is a bit rare for us to find the targetable mutations, and most likely we need a PD-L1 testing to make sure and to assess whether the patient is a suitable candidate for immunotherapy. So this is a therapeutic relevance of molecular classes of lung cancer. So for non-smoker lung adenocarcinoma, usually and very commonly we find the patient will have a targetable mutation. That means it's a single mutation that drives the growth of cancer. So we can block off the receptor with the targeted therapy and control the tumor. For smoking-related lung adenocarcinoma, it's behaved a bit differently because usually the tumor will have multiple mutations. So there is no single mutation that drives the growth of the tumor. That's why they have multiple mutations cause the tumor mutations burden to be high. And this is the group that's probably more suitable to be treated with immunotherapy. Whereas a non-smoker lung adenocarcinoma usually happens in female, targeted therapy will be the main treatments, but we need to rely on mutation testing and also the PD-L1 testing to determine whether what kind of treatment is needed for those group of patients. So if we have this, mutations detected from the specimen, then we treat them accordingly using a targeted therapy. If we can't find any mutations which is targetable, then we rely on a PD-L1 status. So either it's PD-L1 negative, low expression or high expressions of more than 50%. But what is PD-L1? So we'll explain a little bit more later. So molecular subtype of lung cancer. So most of the patients, we can find some mutations, but some of the patients, we actually can't find any targetable mutation. Then we rely on PD-L1 status to give them immunotherapy or chemotherapy. So the landscapes of lung cancer treatment have been evolving very fast. So we used to get one new drugs every probably 10 to 15 years, and now we got lots of new drugs in the markets and in the pipeline, in the research pipeline, and we can target a lot of mutations. And briefly we talk about targeted mutations. So some of the cancer, like what I said just now, lung cancer, they have a single mutation that drives the growth of the tumor. So just one mutation. So if we block off the mutation, then we can control the tumor and we can and then even complete response can happen. So we block off all these receptors. It's like block off the engine. So the cancer cells stop growing and it's a rest. And you go into remissions. So some of the receptor, is there in the cancer cell. But if mutation happen, some of the cancer cell will have a loss of receptor at the surface. So it gives a lot of signal to the cancer cell. Then the cancer cell will grow rapidly and it metastasizes early. So if we block off all these receptor using the targeted therapy, then we control the tumor. But then it keeps accumulating a side effect. So this is a problem without a good solutions. So we keep on blocking, and the cancer can be in remissions for 1 to 2 years, much better than before. But after 1 to 2 years, there's a new mutation that we need to use new treatments or new targeted therapy to block off. But we don't have so many kind of targeted therapy at this moment. Patients probably are suitable for 1 or 2 lines of treatments. Then we have no more medication for that patient, and patient will eventually succumb to the disease. How about immunotherapy? Immunotherapy is a bit different. We know that our immune cell is capable of killing off the cancer cell, but this is not happening. And this is not happening because of one mechanisms called PD-L1 mechanisms. Once a tumor cell bind with the T cell PD-L1 receptor, our cell go into sleep, our immune system go into sleep. But this is the stop and go button. Once we block off the stop button, our immune system wake up, and then we can kill off the tumor cell. Then not all the cell is having this kind of mechanism. We can see that some of the cell is actually quite hot, immunologically hot, especially those cancer which is related to smoking habit and cancer that's related to sun exposure. So all these cancer are related to smoking habit, and this is sun exposure. So it's give rise to hot tumor. Hot tumor sometimes in clinically or in histologically, you can see some of the tumor with a loss of immune cell infiltrations. There's a loss of NK cell, CD8 cell that's infiltrate around the tumor or within the tumor. So when we have all these new treatments and modalities, how do we assess whether this is valid treatments and is better than the old treatments? Usually we measure the overall survival. Let us go and see some of the old trial in the lung cancer. This is published in Lancet 2019. It's a trial for the phase 3 trial for the metastatic non-small cell carcinoma. And this is a trial involving chemo. After chemo, then whether we give a maintenance pemetrexed versus best supportive care. And we can see some of the survival curve here. And this is time. And this is proportions of patients which is still who are still progression-free, and this is overall survival at this time. And we can see the survival curve. The new treatments with the maintenance of pemetrexed as a new chemo is much better than the placebo. And the survival curve, we can see the clear separations of curve, and there's a clear survival benefit at the end of this. So this overall survival difference of 13.4 months versus 10.6 months is about 2.8 months different. And this is a very big breakthrough and these big celebrations. And this drug actually selling like hotcake. So this is a big breakthrough of even 2.8 months increments of survival. But how about now? So if you see the cancer immunotherapy, and this is a classification that I like, and we basically classify the immunotherapy into cellular immunotherapy, antibody treatment like checkpoint inhibitor, or even cytokine therapy. Cytokine therapy is a very primitive forms of immunotherapy, whereby we stimulate the patient's immune system so that in hope that we can stimulate the patient's and host or patient's immune system so they will attack the cancer cell. We know that now this is not a very effective way, but immunotherapy actually started from there. So we look at some of the old trial in 1999. Metastatic renal cell carcinoma knows to be very resistant to treatment. We can give all sort of chemo, but actually we can't create any response rate. So the tumor is resistant to chemo usually. By 1999, scientists start to test the immunotherapy by using interferon alpha, stimulates the patient's immune system so that it can attack the cancer cell, versus medroxyprogesterone. And now that we know that this is a placebo, this probably won't work. And scientists start to see some separations of curve, and progression-free outcome is much better, and survival, then you can see the clear separations of curve at the end. And we start to see some response. This is partial response of about 12%. Wow, this is very good. Complete response of 2% means some of the cancer actually disappeared. And we know that some of the complete response actually become a survivor and the cure from the disease, from the stage 4 lung kidney cancer. And this is a big breakthrough. But the side effect is very significant. And dry mouth, nausea, lack of appetite, tiredness. It's a very significant side effect. Improvement of 2.5 months, and this was a major breakthrough during that time. And subsequently in 2005, scientists tested a high dose interleukin-2 with some others immunotherapy and found that if you give a high dose, the response is even better, 23%, complete response 8.4%. And we know that patients who gets a complete response probably cure from the stage 4 kidney cancer. That is a very, very good outcome. So, but this gives rise to very, very significant side effect like hypotension, 56.8%. It's like anaphylactic reaction, a severe kind of allergy reactions. So, but you can see that those patients who have a complete response or good response, they probably cure for the patients from the cancer, and the result and the response is sustainable. And you can see the survival outcome. There's a clear separations of curve, meaning that about 10% of the patients cure from the disease and they live long term. So whether we can achieve a cure from the cancer with the immunotherapy, and we need to break the patients. Can we have a cure with the immunotherapy without breaking the patients? And now we see the new form of immunotherapy, which might be simple and yet very effective. Do we have something of that? Yes, we can have that. But it's a cellular immunotherapy. That's like CAR T cell, and some other cellular immunotherapy, but it's usually used by hematologists in the treatments of lymphoma and leukemia. But in the solid tumor, we have this sipilosa T as well. It's a kind of cellular immunotherapy that we retrieve the patient's immune cell, retrain it, and infuse back to the body, and is proven to have a better control of a slow-growing prostate cancer cell. But usually in the clinic setting, especially in the lung cancer, we use antibody therapy, and we call it a checkpoint inhibitor. These are the scientists, Dr. Tasuku Honjo and Dr. James Allison, who discovered some of the immunotherapy pathway, like checkpoint, that we can block and have a very good response to the cancer. And these two scientists actually was awarded with Nobel Prize in 2018. One is from Japan and another one is from US. These scientists discovered the PDL1 pathway, and these scientists, Dr. James Allison, discovered the CTL4 pathway. And these are the drugs that used in the clinic, and it's nivolumab, pembrolizumab is blocking the PDL1 pathway. So how it works is actually the drug works by opening the stop buttons in between the tumor and T cell, so that the T cell will start attacking the tumor cell. I mean, you wake the immune cell out so that it can attack the cancer cell. Let us look at some of the trial. This is a stage 4 setting of a lung cancer patients. The Keynote 024 trial is a pembrolizumab is immunotherapy versus chemotherapy in advanced non-small cell lung cancer with a PD-L1s of more than 50% in 2019. And now you can see the clear separations of curve. This is survival curve, meaning at the 24 months, still about 50% of the patients survive from the stage 4 lung cancer. And before this immunotherapy, chemotherapy probably can sustain the life of the patient about 9 months, 7 to 9 months. And now 24 months, still 50% of the patients survive the stage 4 lung cancer. And the placebo chemo is about 30%. But we compare to the trial that we mentioned just now, is a chemo trial. The long-term survival probably is about 10 to 20%. But now we can increase it to 50% for those with PD-L1 status of more than 50%, which who are very suitable for immunotherapy treatments. How about we select patients who have PD-L1 expressions of more than 1% only and treat them with immunotherapy alone? And this is a Keynote 042 trial. It's a phase 3 clinical trial. And now we see the outcome is completely different. As a survival also inferior initially, but after that it's overtake the chemotherapy. And now we know that for PD-L1s of more than 1%, if we treat the patients with immunotherapy alone, probably is not is suboptimal. Even though we know that in long term immunotherapy is better than chemo. So there's an initial drop survival versus chemo. So we are not selecting the patients carefully. What about we use it immunotherapy with chemotherapy? We see the Keynote 189 trial is an immunotherapy plus chemo versus chemo only. Then we see the very well divided survival curve in which there's no initial drop in survival, and the patient is the disease is well controlled with immunotherapy and chemotherapy combination. And this is a trial that makes most of the doctors surprised, because if you give chemo to some persons, a patient actually will depress the immune system, and we give immunotherapy will stimulate the immune system. And in our mind, and this probably won't work, but to our surprise, it worked fantastically. So chemotherapy and immunotherapy sometimes work synergistically for patients regardless of PD-L1 expressions. How about in a curative setting in the stage 3 lung cancer? And stage 3 lung cancer, if you want to cure the patient, either the patients go for operation, and most of the time we give chemoradiotherapy. But after chemoradiotherapy, if you add on immunotherapy, what happens? And this is a trial, a Pacific trial, published in 2018, using the immunotherapy called durvalumab. And we can see the clear separations of survival curve, well separated. This kind of curve never happened before, irrespective of what we do. We try to change the radiotherapy schedule, we try to change the chemotherapy to the better chemotherapy, but it remains just a few months different. But if we add on chemotherapy, the survival outcome is much, much better, up to like 10 to 20% better. So immunotherapy works fantastically in the advanced lung cancer. So more immunotherapy trial in the earlier stage, aiming for higher cure, now is the progress. So, but is immunotherapy applicable for most cancer patients? But actually, no. We see a lot of cases with treatment failure. So we actually need to select the patients carefully. Patient selections is important, and treatment sequencing is important. If some of the patients who are suitable for immunotherapy and we give them chemo first, then probably is suboptimal, because actually immunotherapy works well in the first line setting for some of the patients. So immunotherapy partner is important. Sometimes immunotherapy alone is not sufficient. We need to combine with chemo or some other immunotherapy partner. So financial support is important as well, because immunotherapy actually is quite expensive. So you see some of the immunotherapy related toxicity side effect. So it's just like autoimmune diseases. So sometimes we stimulate the immune system. The immune system overact and give rise to a loss of side effect. And you can see that it's a wide range. It's just like SLE. And sometimes the side effect can be delayed, like happens months or even years later. But overall, this is milder compared to chemotherapy. Commonly seen, we see some pneumonitis, we see some myocarditis, rash, diarrhea, colitis. Hypothyroidism is very common, uveitis, and some myalgias and all these side effects. Usually it's very mild, but some mortality happen of about 0.6% of the patients for a PD-L1 inhibitor. We see some of the trial like Keynote 024 is an immunotherapy compared to chemotherapy, and we see the very encouraging treatment outcome or improvement of overall survival. We can see some of the side effects The grade 3 to 5 side effect for chemo is about 53%, for immunotherapy is about 31%. But you see some of the side effects is usually the immunotherapy related side effects are usually hypothyroidism, which is very, very common, pneumonitis, hypothyroidism or infusion scar reactions, and usually it's quite mild. For some patients with immunotherapy side effect, we treat them with steroid, or even it's more significant, we give them a high dose steroid or even ventilations. And it's even more, and it's can't control with steroid, we give them some drugs for autoimmune diseases like infliximab. So I share some case. It's a stage 2, it's a stage 3 lung cancer. It's my patient, 55 years old Chinese gentleman, presented to me with in 2018 with a chief complaint of left chest pain for 6-month durations. No significant weight loss. Come for second opinion, not keen for operations. So was recommended to have a neoadjuvant chemo followed by radical surgery. But because I'm not keen for surgery, then come to see me. Past medical history, stroke with full recovery. The patient is a chronic heavy smoker but stopped smoking after diagnosis, was treated with Chaldol, it's kind of painkiller. Tumor marker was not raised and weight was 57 kg. Creatinine clearance and kidney function is excellent. Biopsy shows a squamous cell carcinoma. No PD-L1 testing was done because it's not indicated. So in diagnosis, it's a stage 3 lung cancer. So stage 3 lung cancer, if the patient is not a surgical candidate or patient refused surgery, we treat them with chemo radiations. So this is left lung, and this is a tumor, and this is axial view, and this is a tumor. So we give chemo radiations followed by immunotherapy with durvalumab. So 4 months after treatment, pre-treatment, after treatment, it's complete response. 2 years, 10 months post-treatment, and it's still complete response. This patient is totally clear from the disease, and the side effect is only rash, self-limiting. I'm not even give any treatment, the treatment for the side effect. Until June 2021, patient was 71 kg from the initially 57 kg. Means usually it means that the disease is clear, and subsequently patient loss of follow-up. Stage 4 lung cancer. This is the second case. 70 years old gentleman presented with 1 month of cough in 2018, diabetes, hypertension for 30 years, and kidney impairment. This probably not a good candidate for chemotherapy because of the OH, kidney impairment, diabetes, hypertension. Lung biopsy shows this adenocarcinoma, no mutations, targetable, but PD-L1 status of 50%. So we know that this patient is a very good candidate for immunotherapy. And this is the scan of the patient, axial view, and this is lung cancer. And we know that this is another nodule, means it is stage 4 lung cancer. This is a coronal view, cancer here. So the patient was treated with pembrolizumab, a kind of immunotherapy with palliative intent. The disease resolved within a few months. But the side effect is only hypothyroidism, and well treated. So 3 years now, the patient was given 2 years of immunotherapy, and the 3rd year, there's no evidence of disease. The disease is completely gone. And I last saw the patient in January this year, the disease is still clear. The patient is still well. Now the main concern is the diabetes control. So in conclusions, immunotherapy potentially creates a long-term survival and sometimes curing a stage 4 lung cancer patients by changing the defense system of the body without causing excessive side effect. But as patient selection is very important, and we need to select the treatments well as well, because some of the patient might need immunotherapy plus chemotherapy. And we'll accept some Q&A after this. I think I'll pass it back to Chairperson. Okay, thank you very much.
Thank you, Dr. Ivan, for the interesting talk. I believe Dr. Ivan has managed to join us, so we can actually pose some of the questions to him live later on during the Q&A sessions. So once again, if you have any questions, please post into the chat box. Yeah, we're going to moderate them very soon. So now we are moving to our very last speaker of today, but definitely not the least. We have next will be with us is Mr. Narasimhan Satyamurthy. Just hang on, let me just get back his CV. So for everyone's information, Mr. Nara is currently working in the Department of Surgery, HKL. He is the consultant thoracic surgeon. He's also the head of the unit in HKL. And besides that, he is also holding the positions of head of the thoracic surgery service in Malaysia. Mr. Narasimhan actually has also, following his master in surgery, he has done his fellowship with the Ministry of Health Malaysia, and also a year in the Department of Robotic and Thoracic Surgery in India. Mr. Narasimhan actually has published extensively, and he has also spoken in numerous conferences, both internationally as well as nationally. So after listening to Dr. Ivan Shiu regarding the medical approach of lung cancer, now let's just change gear to the pulmonary approach, or surgery approach, in lung cancer. Mr. Narasimhan.
Hi, thank you to the Madam Chairperson for the kind introduction. And after listening to extensive discussion on the molecular basis of lung cancer and the adjuvant and immunotherapy targeted treatment, perhaps now maybe you can just spare the last few moments of this talk listening to the surgical aspect of lung cancer and how is it like in Malaysia. We've listened to the previous speakers that lung cancer usually presents late. So what can be actually done in our country? Well, our population is about 32.5 million, and we're covering an area of about 330 kilometers squared in Malaysia. If you look at this distribution of non-communicable diseases in Malaysia, one of the first malignant disease that appears as the #1 killer is lung cancer. The rest of this is all non-cancerous, non-communicable diseases. And so that is something to be worried about. Lung cancer is among the top malignancy that kills Malaysians. The rest of the cancers falls at the bottom part of this chart. So there is a very important reason why we need to address lung cancer in Malaysia very seriously. And I think it's been long overlooked in terms of the surgical aspect of it. So this is the statistics we have, the common cancers in our country, and it's third commonest after breast and colorectal. But if you look at the mortality, although it's third commonest, it holds one of the highest cause of mortality in this country. It's #2 after breast cancer. And this is the presenting stages of lung cancer. Most of the patients, they present in stage 3 and 4. Rarely we get stage 1 and 2. It was discussed by our previous speaker. But more importantly, it's good to look at the what we call the 5-year survival rate of lung cancer in our country. And if you notice, the survival drastically reduced from stage 1 to stage 4. And later we will compare the survival with our neighboring country, and we will discuss on why. And this is the 5-year survival of lung cancer if you compare with other malignancies. And whereas lung cancer, it's right at the bottom of the chart. So it has got the worst survival rate if you compare with other malignancies. And if you compare with other countries, this 5-year relative survival, Malaysia is somewhere in the last 3, just above Thailand and India. And we will discuss on why our country is doing so badly in lung cancer survival, despite being the #2 killer in the country, and what is the reasons behind this. So this is my first summary. Is the third commonest cancer diagnosed in our country. Is the commonest cancer-related cause of death. Almost 90% of lung cancers diagnosed stage 3 and 4, and it's got the worst 5-year survival. So I hope you get this in context. So what can be done? I'm showing back this chart. Malaysia is somewhere the last 3 worst survival, but let's look at China. China has got huge population, and they got among the better survivals in this region. If you look at the population of China, is about 1.4 billion people, 28% are smokers. That means 397 million smokers are there in China. In Malaysia, the number of our population is less than the number of smokers in China. So it's 32 million. The percentage of smokers is about more or less same, 22%. And we have about 7.4 million smokers. So in China, nearly 1 in 3 smokers in the world is Chinese. And in 2018, there were nearly 800,000 new cases of lung cancer diagnosed in China. And every year, over 1 million Chinese people die from tobacco-related diseases. So despite all this, how can they have good survival for lung-related malignancies? And this question has been toying around in my mind for a while. So in Malaysia, if you look at it, we have 23% of male in Malaysian dying of lung cancer. It kills about 403 men every week, necessitating action from policymakers. So this is what's the situation in Malaysia. You have people dying, but not much has been done to prevent this kind of death. The ironic part is it is preventable. How? So let's look at the 5-year survival comparing China, AJCC, and Malaysia. So, okay. So this is the, I'm not comparing AJCC, just compare China and Malaysia. So this is a Chinese multi-institutional registry for resected non-small cell lung cancer on 5,853 cases. And this is back in 2013. Stage 1A, 1B, compared to Malaysian study on cancer survival 2018, 4,715 cases. They have 81.9% stage 1A and stage 1B, 71.6% survival. Compared to Malaysia, 37.1%. It's not even half of their survival. We don't have 1A, 1B data. Look at the subsequent stages. And you can derive yourself why. You have to ask yourself why you are doing so badly. This is simply because perhaps we're not detecting them early enough. And what is China doing differently than what we have? So I visited Shanghai myself in 2019 to try to find out what's going on. And I visited Shanghai Pulmonary Hospital. It is a dedicated hospital for lung diseases, lung cancer mainly. They are the largest center for thoracic surgery in the world. They perform about 15,542 lung cancer surgeries in 2018 alone. This is not lung cancer patients. These are lung cancer patients undergoing surgery in that center. So, I presented our part of our experience in Malaysia, and it's nothing compared to what they've been doing in China. So it's the largest thoracic center in China. It's government run. They do about 90 to 100 lung resections a day for malignancy. They perform low-dose CT scan for their population. It costs about 120 to 160 ringgit. They do not have any lung cancer screening program. The patients pay up to 20,000 ringgit for lung resections in government. Some of them are subsidized and insured. Those who can afford who are not subsidized, they pay cash. The awareness of the public in Shanghai is excellent. They're concerned about their malignancies, especially lung cancer, knowing they have the highest percentage of smokers in the world. And then later to find out the EGFR positivity among the Oriental people is as high as 30%. So they're quite aware and they go for the low-dose CT scan. In HKL, that's where I work. In the last 5 years, we've performed about 1,000 thoracic surgeries for various indications. And sad to say, we only operated on 64 lung cancers. That's what, 6.4%. And if you compare to Shanghai Pulmonary Hospital, more than 80% of their surgical load is for lung cancers. And this is the breakdown of our patients based on stages. Stage 1 got 16%, 39%, and most of it is 45%. And we get these stage 1 patients on a random basis when they go for medical checkup, incidental finding of some small nodules in the chest X-ray, which is investigated, and that's how they end up to me. And this is over 5 years. I have to emphasize The 5 years period, we only operated 64 patients. That's very sad. And why is this happening? Awareness. So most of these patients will say, I've been smoking since 15 years old and I'm still fine. Why bother checking? That is the attitude we have in our population. They just don't bother to get themselves checked. Even my own father-in-law is a smoker, and it's difficult for him to stop smoking, but we have persuaded him to go for low-dose CT scan, and God's grace, it's fine. And he said, look, I told you it's fine. Why bother checking? And now he refused to go for a routine checkup. So this is a problem with the population in Malaysians. The expertise. We only have 3 thoracic surgeons in government service. There are only 6 thoracic surgeons registered in NSR. And the other 5 is either in the universities or private practice. And not many are doing radical surgery for lung cancers. So that is a problem, and we are training now. Infrastructure. We don't have a dedicated chest center in the country, but I'm happy to say it's currently in the planning phase in combination with the National Respiratory Center. So we are in the process of getting our own national chest center. So the motto should be detect early, treat early, you get better outcome. And how to detect early is you have to identify patients who are high risk. So the high risk patients, mainly male, more than 50 years old, and if they are more than 20 year smokers, female, Southeastern Asian, non-smokers, family history of malignancy, counsel them regarding the benefits of performing LDCT, low-dose CT scan, and enlighten them regarding the risks of getting lung cancers if they fall in the risk categories. And also explain to them about Nelson trial, which is and Talent study, the Taiwan study, which concluded that early detection saves life, and direct them to the hospitals offering LDCT, which can be either performed with subsidized cost, or some hospitals do offer them for free. So this is a Nelson trial, and it is a very large randomized population-based trial confirming the value of low-dose CT scan. And it shows that CT scanning decreased mortality by 26% in high-risk men and up to 61% in high-risk women over a 10-year period. Screening in Malaysia, it was discussed in our health technology assessment report for the role of low-dose CT scan in lung cancer. And it's been concluded that LDCT can only be used for lung cancer screening among the high-risk group in a research environment or for research purpose only, because the cost-benefit ratio of performing a low-dose CT scan, it's not justifiable based on this assessment. So we cannot proceed with lung cancer screening in this country based on this kind of evaluation. So we have tried to perform lung cancer screening pilot program. It was started in IKN. It failed. It was not, there are not enough takers to come forward and offer themselves to be screened. We attempt to collaborate between NGOs, private stakeholders, and government institutions for screening. At the moment, it's failing. Government hospitals are overburdened and understaffed to perform LDCT as medical checkup or screening. We can't even meet the requirement to perform our usual scans. The waiting time somewhat goes to 1 to 2 months at the moment. So what is being planned in MOH? The infrastructure is in need of a one-stop center for chest diseases. As I mentioned earlier, it's in the planning. A location for low-dose CT scan to be performed in government hospitals, still in the discussion, since the health technology assessment has proven that it's not, there's no cost-benefit ratio in performing LDCT. We are training more thoracic surgeons to perform radical surgery for lung cancer. And awareness is to be made widely known the benefits of detecting lung cancers early. Early diseases are almost always asymptomatic. So the only way to detect them is by performing LDCT, and it has to be performed in the pretext of medical checkup. And what can the public do? They have to equip themselves with knowledge, they have to read, invest in their health, include LDCT as part of the medical checkup, and the hospitals must reduce the price of this CT scan so that it is made affordable for any public to go walk in and get themselves checked if they fall in the high risk category. The primary care physicians play an important role. Look for patients with high risk to get lung cancer and not look for the symptoms. They are almost always asymptomatic to start with. Advise them to perform LDCT. Identify the hospitals that can perform LDCT for free or discounted price. Review the scan results. Don't make decisions to follow up on yourself, and refer to thoracic surgeons or respiratory physicians early. So these are the places where the LDCT is being offered for free or in subsidized manner. I'm not sure if they still have it, but this is in 2020 they had it. Hopefully we do that again sometime this year or next year. So this is my second summary. LDCT is a safe way to detect early lung cancer. Include LDCT in medical checkup. Consult respiratory physician or thoracic surgeons early. Private hospitals should encourage LDCT to high-risk patients to come for medical checkup. Now let's talk about the surgical option. This is the last part of my talk. We have early lung cancer, advanced lung cancer, metastatic lung cancer. So early lung cancer, stage 1A to 2B, the options normally surgery, and we can perform VATS lobectomy, lymphadenectomy, VATS sleeve resections if it is in the common bronchus, secondary carina. And for stage 3A, advanced lung cancer, if it's single station N2, that means if you have a single station mediastinal node, that's based on the staging of lung cancer. You can perform VATS lobectomy and lymphadenectomy upfront and then followed by adjuvant treatment. Or you can give a new adjuvant treatment, surgery, and continue with the adjuvant treatment, depending on the pathology results. And if they have multi-station N2, most of the time it end up with chemoradiotherapy with TKI or immune checkpoint inhibitors. Metastatic lung CA has been extensively discussed earlier, and is chemotherapy, radiotherapy, targeted treatments, and immunotherapies. So this is the conclusion from Violet trial. It's a randomized trial to compare video-assisted thoracoscopic surgery versus open thoracotomy in patients with early lung cancer. And it is a pleasure to inform that VATS lobectomy, minimally invasive approach for early lung cancer, has been decided as a gold standard approach to treat early lung cancer surgically after these trials results were announced. It's proven to be less painful, significantly lower in-hospital complication, shorter length of stay, and these were achieved without compromise to serious adverse event and oncological outcomes if you compare VATS and open surgery. So in this day and age, most of the thoracic surgeons must offer VATS as an option, surgical option to patients who presents with early lung cancer, because it's proven in our RCTs. So this is a surgical example of a right lower lobectomy and lymphadenectomy in a patient with stage 2A lung cancer, which we perform in HKL. There are actually 4 stages of this surgery. You have to first dissect and ligate and divide the inferior pulmonary vein, then move on to the basilar artery, then we remove the lower lobe bronchus and then perform lymphadenectomy. So let's go one by one as I guide. And this surgery was performed in the right uniportal VATS, means it's a single incision VATS surgery. The incision is made about 4 centimeters, and all the surgery is performed through that incision alone. We don't have any other incisions. So this is how the surgery is performed. You have an energy device to dissect, and here you can see the inferior pulmonary vein being dissected away. And we divide this using a parenchymal stapler. Traditionally, the morbidity of an open thoracotomy scares the patient as well as many physicians, even to offer this surgery to patients. But nowadays, with this minimally invasive surgery, especially uniportal VATS surgery in the limelight, we can perform surgeries in patients with who even have some comorbidities, who are older patients, and offer them curative resection. So this is the vein has been divided. Now we move on to the artery. So this is the basilar artery, and the basilar artery need to be dissected very carefully and delicately. It is more fragile than the vein. Any injury to the basilar artery, it can be catastrophic. It will fill up the thoracic cavity within 30 seconds and patient can collapse. So the dissection must be done very delicately, precisely, with good knowledge of the anatomy, and hence you need to be addressed by a well-trained thoracic surgeon. So this is the basilar artery that's supplying to the right lower lobe of the patient. So we dissect it around, make sure the sheath is cleared, and then we encircle the basilar artery and divide it again with a vascular stapler. And in this day and age where we have improved surgical devices like the staplers that you've seen me using, these surgeries can be done within 2 hours with good lymphadenectomy. And the patient will be extubated immediately after surgery, and they can be up and about and walking within hours, 6 to 12 hours after surgery in the ward. They'll be allowed orally and they'll be home within day 3 of surgery. So this is how we have evolved over the years from the conventional open thoracotomy, rib resection and so forth, to only performing surgery with a single incision, 4 centimeter in length, and the whole surgery is completed through that. So after this artery is divided, we move on to the bronchus. So this is how the bronchus looks like to the lower lobe, and this above it is the middle lobe bronchus here, you can see. So you need to be careful not to injure the middle lobe bronchus. You have to dissect clearly and cleanly, make sure there's no nodes sitting at the bronchus, and is again divided with a parenchymal stapler, carefully without injuring the middle lobe bronchus. And finally, we move on to the lymphadenectomy. So the lymphadenectomy, normally I'll remove station 7, station 4, station 2, station 9, depending on the nodal status of this patient preoperatively. So in this patient, the N2 nodes were negative, but there were a lot of nodes seen, so I removed them anyway. Radical lymphadenectomy is performed, and there are clear landmarks to achieve when you perform this surgery without injuring the esophagus behind here, the vagus nerve, the carina. So again, the dissection must be done carefully. This is going ahead and performing station 4 and station 2 lymphadenectomy. And during this surgery, we need to expose the trachea. The brachiocephalic artery has the floor. The SVC will be anterior to the dissection. And need to be careful not to go too high because you'll injure the recurrent laryngeal nerve on the right side. So this surgery have to be done based on strict anatomical landmarks, limiting the dissection when we achieve these landmarks during the surgery. So now I'm doing the last bit of the lymphadenectomy towards the apex of the R2 station, and this is the branch from the vagus nerve coming into the node, and that will be the end of my dissection of the lymph node. So once that is removed, you create a crater here. The anatomy must be revealed: the vagus, the trachea, SVC, the azygos, and posterior. The floor will be by the brachiocephalic artery. This is the azygos vein. So that is the end of the whole thing, and this is the lung that's removed, and this is the wound. You perform the whole thing, and the drain is even inserted in the same wound, so the patient will not have any other surgeries. So in conclusion, lung cancer is the commonest cause of death in Malaysia. The 5-year survival of lung cancer is worst. Early detection of lung cancer saves life. LDCT can be performed as part of medical checkup. VETS lobectomy is a gold standard in the treatment of early lung cancer. And with that, I thank you. If you got any questions, we can discuss during the Q&A session. Thank you.
Thank you again to Mr. Narasimha for his excellent and wonderful talk and sharing. Could we invite back all the speakers perhaps to on the camera so that we can actually move on now to the Q&A sessions. We actually have quite a number of questions in the chat box, so hopefully we can clear them before the next session, which is the very interesting real case presentations by our medical students. Sorry, okay. All right, perhaps I can start off with the first questions. The first question is actually to Dr. Wang. Dr. Wang, you're with us, right? Okay. So the question is, does other circular RNA, for example, 103820 suppresses lung cancer tumorigenesis?
Regarding circular RNA, I believe that there are lots of circular RNA that are gradually being discovered to either to promote lung cancer progression or to suppress lung cancer progression. Actually, in our lab, we also, from our NGS data, we have several candidate circular RNA that may directly or indirectly regulate the progression or the migration or metastasis function of lung cancer. So that is, yes, that there are lots more circular RNA waiting to be discovered. I can hear your voice.
Sorry, I've muted myself. So the second part of the question is there any recognized useful specific circulatory RNA as prognostic indicator?
Actually, there are some paper reporting that identifying circular RNA from cancer tissue could be a potential marker, or using just a few paper reporting that using circular RNA as a liquid biopsy marker. From my point of view, liquid biopsy marker will be more critical for the future clinical use because you can easily monitor along with for example, your treatment course, that you don't need to do another surgery to know that the status of this circular RNA expression status to reflect the disease, the current status of it. So actually, if you read the literature, lots of papers still state on the correlation study about circular RNA and certain disease, not even cancer, but also, for example, some neurodegeneration disease, the correlation. But not really lots of paper trying to understand, is it just a marker or it also involved in some pathogenic pathway of that disease? I think that's some of the hurdle in the field of circular RNA we need to overcome.
Yeah, thank you.
Thank you, Dr. Wang. There is one question maybe I can invite Dr. Nora Shikin. Dr. Nora Shikin, may I know that are there any differences in the lung cancer stem cells isolated from different model? Example, 2D cell culture, spheroid or organoid?
Dr. Nora Shikin, you are muted.
Okay, there definitely a difference if we are isolating cancer stem cell from, for example, the monolayer 2D cell line, or by using spheroid. So in our lab, when we grow the cancer stem cell in 3D model, we can see that the cancer stem cell that we that we isolate from spheroid, they are more resistant towards certain therapy as compared to the one that we grow from 2D monolayer culture. Okay, that's one of the main difference between these different model. And as for organoid, so because this is still new and we did try to grow cancer stem cell into organoid model, and it shows that it will give a better representation of the what we call that, the complexity within the tumor as compared to the 2D and also the 3D spheroid model. Is that answering your questions?
Yes, Dr. Norashikin, thank you for your answer.
Dr. Shikin, perhaps I could just ask you this question as well. Do you think in the future we can actually use cancer stem cells as a prognostic indicator to monitor relapse of patients with cancers?
Okay, so actually, in my opinion, I think yes, because there is research showing that cancer stem cells can be considered as what the metastatic cell that they are, what we call that, circulating. So these cells are actually the cells that migrate from the original tumor into the new regions of the organ. So if we can detect the marker or the biomarker of this cancer stem cell that circulate in the bloodstream, maybe we can use them as the maybe a diagnostic or prognostic marker for lung cancer.
Okay, all right, thank you, Dr. Shikin. I have one question here to Dr. Wang. Can we isolate circular RNA from other bodily fluids other than from the blood? And if yes, what are the yield like compared to the blood? Dr. Wang, you're muted.
Sorry. Actually, we haven't tried on any other body fluid so far, because when we start to try to isolate the circular RNA 190 and 1649 from the patient's plasma, we did test lots of different protocols. So it's not an easy way to isolate secreted circular RNA in body fluid. And I think the blood is the more direct part that the secreted molecule could be exist from patient sample. And other parts, people would some paper would suggest that, for example, saliva or urine, but I haven't seen very solid data that people isolate circular RNA from that.
Okay, thank you again, Dr. Wang.
You're welcome.
Moving next to the questions, I think this is directed to Dr. Ivan. Dr. Ivan, the question is about CAR T. What is the role of CAR T in lung carcinoma, and which type particularly would you consider CAR T therapy?
I think CAR T is still very investigational in lung cancer. There's no good data that supports CAR T in the clinical use. So I think CAR T cell is more suitable for hematological malignancies, and then more data is over there. But the CAR T cell, I think probably this kind of techniques probably will be a good modality of treatment in the future, but probably not now. We have no good data yet at this moment.
Okay, all right. Perhaps I could just ask you one question as well. Where can we actually do the mutation analysis in Malaysia, as well as the PD-1 expression? Do we send to the IMR lab or a private lab?
I'm not too sure about government setting, but private setting, usually we can send it either to University Hospital or Pantai Lab or Subang Jaya Medical Center. These few major labs. Sometimes we even send to overseas. But nowadays we do all these mutation tests in a broad, how to say, it's what we call is a next generation sequencing. That means we do a sequencing of multiple genes, about 50 to 100 genes, so that we can get back the result faster. So either we send overseas or locally, but usually nowadays we send locally because we can get back the result very fast. So it's called next generation sequencing. So we sequence a lot of genes, and at the same time, then we scan for the PD-L1 status of the patients. So local lab usually.
Okay. Can I know the cost roughly, like if the entire panel?
Cost is getting more and more affordable. I think now it's about 1,000 to 2,000 ringgit Malaysia only. It used to be about 6,000 to 7,000. Yeah. Okay. The turnaround time is getting faster and faster. Last time we used to wait for around 3 weeks. Now it's about 5 to 7 days.
Oh, all right. That's fast actually.
Yep.
Yeah. So meaning, do you have patient selection who will go for that analysis, or all your patients you actually subject them for that?
I think lung cancer who are not, I mean, small cell we won't send. Non-small cell, I think now the guideline that we will send everybody for this NGS and PD-L1 testing. Even for squamous cell, because last time the thinking is we only send non-squamous for NGS, but because sometimes there's a concern that they might be squamous and non-squamous component. When you biopsy, you just get the squamous component, but actually the patient is having some mutations. But usually all this mutation is, happen in non-squamous lung cancer only. Sometimes for squamous also we will send, especially those who are like adenosquamous kind of histology, or female patients, non-smoker getting a squamous carcinoma, then we will send for all this mutation testing.
Okay, thank you. One more question to the Ivan. Can I just clarify my doubt? Just like you mentioned about the PD-1 expressions, like let's say very high, more than 50% and more than 1%. Is it true that if more than very high, more than 50%, we can actually use the monotherapy of the anti-PD-1?
Yeah, current guideline is if PD-L1 is more than 50%, then immunotherapy is good enough to control the tumor. So for those in between 1 to 50, that immunotherapy probably is not sufficient. So we need to combine chemo with immunotherapy, both. So unless a patient is not a good candidate for chemotherapy, then we will use immunotherapy alone. If not, then we will select patient PD-L1 more than 50%, then we will give immunotherapy. 1 to 50%, then we will give chemo plus immunotherapy. For those who have no PD-1 expression, but they have no mutation as well, then we will give immunotherapy plus chemo as well. It's proven to work.
Okay, despite the PD-1 expression is negative, right?
Yes, but provided the patient is mutation negative. For those who are mutation positive, there's no role of immunotherapy actually.
Okay, all right, thank you. Moving on, I think all these questions are for Mr. Nara. Mr. Nara, all right. The first question is about the VAC surgery. Actually, I'm too very surprised when you share with us there's only 6 registered NSR cardiothoracic surgery in Malaysia right now. So
sorry, not cardiothoracic, it's thoracic.
Thoracic, okay.
Yeah, we have quite a number of cardiothoracic surgeons in the country.
Yeah. So a lot of them actually major in the cardiac side rather than the thoracic side. Yeah. So I think Mr. Yan would like to know about the challenges, moving forward, training the trainers. Training of the trainees, and what do you expect? How to improve on this regarding the training of the new specialists.
As I said, the importance of thoracic surgery has been long overlooked. This field has existed in the country officially in NSR for about 20 years now. But unfortunately, not much importance were given because most of our lung cancer were detected in an inoperable state. And when these patients come to us, especially in stage 3A, most of us decided that it's better to go for chemo radio rather than surgery, owing to the morbidity of the open thoracotomy. But in this day and age, when most of the surgeries are done minimally invasive manner, the thoracic surgeons have to take up the challenge to learn minimally invasive method in treating lung cancers, which is proven by randomized trial. It's got lesser morbidity, lesser complication, and equal oncological outcome as how it was before. So the training is slow. We've already completed training for 2 of my trainees. They've completed their complete a 3 years training, and we have 2 other trainees at the moment. And it is a slow process, and we have to be patient, and hopefully the attrition rate to the private service is lesser. They stay on in the government and continue the service. And I hope the public-private partnership will be established better by the MOH so that most of our surgeons can work both in private and public, and the expertise is not lost to the lesser privileged public in Malaysia.
Yeah. Do they need to go through the Master of Surgery through Mr. Nara
Oh
before?
yeah, all those who are in a thoracic general surgical program have to be general surgeons NSR registered and have completed at least 2 years of service as general surgeons, and then they are allowed to come into the training program for thoracic surgery.
Yeah. Okay.
Yeah.
Thank you. And Mr. Nara, there's more questions here. For the early stage of lung cancer, after the surgical approach, for example, lobectomy, is there any role of adjuvant therapy like radiotherapy, or would you consider some patients for low dose, short course of chemotherapy, or just the lobectomy is considered?
Well, that depends on the stage that the patient falls into. If is 1A, if it's stage 1A, which we rarely get, then if the margin is cleared and all the lymph nodes are not involved, then it depends on the oncologist, and most of the time they would advise there's no need for adjuvant treatment. But in the advent of this immunotherapy for early lung cancers, there is a role to offer these patients immunotherapy to make sure that they have a long disease-free survival. Perhaps our friend can explain on that later on the role of immunotherapy in early lung cancer. It's quite a good discussion going on in the international scenario. In terms of stage 2A, 2B, definitely there is a role for chemotherapy if the patient wants to go for adjuvant chemotherapy, as well as the targeted treatments. In terms of for stage 3, there's no doubt. Normally, the oncologist will advise the patient to go for adjuvant treatment. Normally, I'll leave it to the oncologist to decide on what is the best approach of adjuvant treatment for this group of patients. For radiotherapy, is solely dependent on the margin of resection. If the margin is clear, the lymph nodes are cleared, there's no need for radiotherapy. In some situations where the chest wall is involved and we did not resect the chest wall completely, we just shave it off, then yes, we give some radiotherapy to the chest wall. But otherwise, if the surgery is done well with good lymph node clearance, there's not quite indication for adjuvant radiotherapy for these patients.
I see. Yeah, thank you, Mr. Nara. Dr. Ivan, would you want to share something regarding the early use of immunotherapy for early stage?
Early use of immunotherapy is still, that there is actually phase 3 trial that's recently published showing that there is benefits of giving immunotherapy for patients with lung cancer. But the trial for us is still a bit early because we only see the disease-free survival benefit at this moment. Yeah. So we'll select patient carefully. Usually we need to show that there is overall survival benefit. Means we will improve the patient's survival. Then only we can adopt this kind of treatments. But there is, it's in the research setting, and I think that some of the patients, especially those with a PD-L1 high and chronic smoker, patient who are a smoker, then we can consider immunotherapy at this moment. But usually we'll explain to the patient it's still early, development.
Okay, thank you to both. Is there any more questions from your side, Dr. Ong?
No more questions from my side.
Okay, thank you. I think I have none as well. So thank you again to all the speakers for answering all the questions, and of course the lectures as well. It has been a wonderful half of the sections. Next, we move on to the next sections, which is the case presentations. And this case presentation will be presented by our two beautiful ladies here. They are our year 5 medical students, Jashin as well as Regia. And of course, again, we have our the panelists. Yeah, we all knew Dr. Mr. Narasimhan, Dr. Ivan, and let me take the opportunity to introduce to everyone Dr. Dean. Dr. Dean is actually with us at Utah University. He's the radiologist who's also currently working in Ampang Hospital Radiology Department. So he would join Mr. Nara and Dr. Ivan as the panelists for the case discussion. So may I invite the presenters?
Sure, Dr. Lee. Let me just arrange the screen for sharing first. Thank you.
Hey, good morning, everyone. I'm Rija. I'm a fifth-year medical student from University Tunku Abdul Rahman. So my colleague Josh and I will be presenting a case of non-small cell invasive adenocarcinoma of the lung. And we're also very lucky to have our panelists, Mr. Narasimhan, Dr. Ivan, and Dr. Dean together with us today. So this patient, Mr. M, is an 82-year-old gentleman from Myanmar who previously worked as a bank manager. And he came with intermittent right scapular pain for 2 weeks. So in November of 2021, Mr. M complained of intermittent right scapular pain, which was not relieved by over-the-counter analgesics, and it was associated with unquantified loss of weight. He then presented to a local general practitioner and was referred to a specialist due to abnormal lung examination findings. He has history of smoking of 5 sticks per day for 20 years. Otherwise, there are no respiratory symptoms, no other constitutional symptoms of malignancy. Mr. M has no family or personal history of malignancy or similar conditions. On physical examination, Mr. M generally appeared to be thin and emaciated, and there was no chest expansion over the right lung, no tracheal deviation, no air entry over all right lung fields, and on percussion, there was stony dullness over right lower lung zones, and there was a single fixed and large hard right supraclavicular lymph node measuring 2 cm, and there is no organomegaly.
Good morning, everyone. I'm Jashin. So I'm the second presenter of the day. I will continue on the subsequent investigations and procedures done for this patient. So this is a brief summary of all the positive findings in the laboratory investigations. So on full blood count with white cell differential, Mr. M had a hypochromic microcytic anemia with a neutrophil predominant leukocytosis. And on blood urea and serum electrolytes, he actually had hyponatremia, hypocalcemia, and hypochloremia. Liver function test also revealed that there was hypoalbuminemia, raised ALP levels and GGT levels. There was also a raise in the C-reactive protein levels. So moving on to the imaging, I would like to pass on this part to our expert panelist, Dr. Din Shinjet, for further discussion.
Hello, thank you so much. I would like to share my screen.
Sure,
Can everyone see my screen?
Yes, doctor.
Okay, good. Okay, so this the patient has gone for a chest X-ray as part of the evaluation, but of course, this view, the CT scan and the chest X-ray that I'm going to show is a little bit suboptimal because these are not the real DICOM images, but more of like images taken from the handphone camera. So the resolution will not be that good. So, but anyway, so now we just have a look at the chest X-ray. So when you look at this chest X-ray, the most striking thing is that, of course, you can see that the right lung field is filled with this homogeneous opacity. So this homogeneous opacity occupies the lower, mid, and part of the upper zones. Only a small portion of the apex is left aerated. And at the same time, you can see that this opacity obscures the right heart margin, the right hemidiaphragm, as well as the costophrenic angle. Okay, then you look upwards, you can see there's a presence of meniscus sign. So this is a large right pleural effusion. But when we see such a large pleural effusion, I would expect it to have a little bit more of a mass effect. But when you look at this chest X-ray, you can see that the mediastinum and the trachea is not that much deviated to the contralateral side. So if that's the case, I would postulate that there might be some form of collapse of the parts of the lungs, right? So you have a large pleural effusion with lung part of lung collapse. Then I'll bring your attention to this area. So have a look at the trachea. So you have the trachea, right main bronchus and left main bronchus. And this line is what we call the paratracheal stripe. This on the right side, and this is the left paratracheal stripes. Okay, so the right paratracheal stripes is the interface between trachea and the left upper lobe, as well as the pleura. So you have a little bit of fat within. So usually in a normal person, the paratracheal stripe is actually very thin. Not more than less than 5 mm actually. But when you look at this patient, you can see the paratracheal stripe is very much thickened. So you can see this whitish soft tissue lesion, soft tissue density at the right paratracheal stripe region. So what could that be? So you have to know at the trachea region, there's actually a lot of lymph nodes. So when you see widened right paratracheal stripes, so most of the time it's due to lymph nodes enlargement. So probably patient has a lot of paratracheal lymph nodes. And of course, there can be some other things. Sometimes pleural effusion, when the fluid go to that area, you can also have widened paratracheal stripes, as well as fat lesion like lipoma. But of course, lipoma being fat, usually the density is a little bit lower. So this is the right side. Left side is a little bit hard to comment because in a normal person, even the left paratracheal stripe is a little bit thicker compared to the right side because of there are some other structures there like the subclavian artery. So in conclusion, this chest X-ray shows a very large, massive right pleural effusion with probably some form of lung collapse with multiple paratracheal lymph nodes. Okay, so now we go to this CT scan. So this CT scan is a contrast-enhanced CT scan. Usually we look at the two main windows that we look at is the mediastinal or the soft tissue window, mainly to assess a lesion, large lung lesion, the surrounding structures such as the vessels, lymph nodes, muscles, right? And the other window would be lung window, when we mainly assess the lung parenchyma to look for nodules. Okay, so this is the mediastinal or soft tissue window. We go from up to down. So this is at the lung apex. Okay, so even at the lung apex, you can see some so here you can already see this pleural effusion right until the apex with a little bit of aerated lung apex, right? Sorry, before we go further, I just need to show you a little bit of the anatomy. So here in the middle is the trachea, still relatively centrally located. Besides, you have the thyroid, and behind this is the esophagus, and of course a lung apex. So I bring your attention to this thing. This is a supraclavicular lymph nodes. As you can see, it's adjacent to the clavicle, the right clavicle. So you have a right supraclavicular lymph nodes. So as we get lower down, you continue to see all these pleural effusion. And there's a soft tissue mass, not very clear because of the posterior and anterior segment of the right upper lobe is also partly collapsed. So with this kind of condition, so any lung lesions will not be very well visualized, but still you can see there's some lesions here. And when you look at this lesion, the margin between this lesion and the intercostal muscles is not very clear as compared to the back side. Here you can see, even though there's a big pleural effusion, but you can still see the margin, but not so much here, right? And at the same time, when you look at the pleura appears a little bit thickened and hyperdense with irregularity, right? So you have a lesion here with thickened pleura and also not very well defined margin with the chest wall. So I would like to have a bone window to better assess if the lesion has causes destruction of the rib. Then I'll bring you back to the middle part where you can see a paratracheal lymph nodes. So remember, I told you about the right paratracheal stripes. So this is the cause of the thickened right paratracheal stripes. So you have multiple. As we go down, you'll see even more right paratracheal lymph nodes. So same thing here. This is the lungs. part of the upper lobe is collapsed, especially the posterior and anterior segment, with a very big mass here, large pleural effusion, and paratracheal lymph nodes. Okay, same thing here, multiple lymph nodes. So this is the carina region. Probably there's a carina nodes. We usually will look hard for the carina nodes because it actually matters in the carina nodes involvement will actually change the part of the staging. And here you can see this part of the lungs that goes anteriorly is likely the middle lobe, right middle lobe, and this part is the posterior, the lower lobe. So both has collapsed. So that's why there's not much mass effect, even though there's such a big pleural effusion. Okay. And this is the last slide where you can still see the pleural effusion with part of the abdominal organs. But sadly, this is only a thorax CT scan, so we don't get the full picture of the abdominal organs, so can't really comment much on the metastasis. But here, from what I can see, there's no evidence of metastasis to the liver as well as the kidney, the upper abdominal region. Okay, now we go to look at the, this is the lung window. So lung window, right side, not much you can see because just a very small portion of the lung remains aerated. But on the left side, the left lungs looks relatively clear. So you don't really see satellite lesions or any nodules on the contralateral lung. This is from what I get from the images, the WhatsApp images, okay? Okay, now we go to the staging. So when we see something like this, most likely it's going to be lung tumor. And I had the pleasure of working with a chest thoracic radiologist when I was doing my master in HKL. So my the thoracic radiologist would usually advise us to stage the case, even though we don't really have confirmation that this is a lung cancer. We just if you suspect it's a lung cancer, we will advise just to straight away stage it using this DNM staging, because if it's a lung cancer, of course, the clinician will be able to look at your staging straight away, other than we wait for confirmation and then they may have to send the images back to you to restage, or they have to stage it themselves. So it'll be quite hassle for the clinician. So we are advised to do that. However, I think some, if not most, of the radiologists don't feel very comfortable or confident in doing that, giving a cancer staging before confirming that it's a cancer. So yeah, so that's the usual case. And here I have two editions of the DNM staging. I'm not sure how the 8th edition is the more recent one, but I'm not very familiar with the 8th edition. I'm more before this we use the 7th edition, and as you can see, the 8th edition is a little bit more complicated. You have more subdivision, right? But anyway, I'll try to stage this patient by using the 8th DNM staging. Okay, so first is we look at the size of the lesion. So here I can just agak-agak, so I can't really give a confirmation how big is the lesion, because we are supposed to look at the CT scan and adjust the plane and then measure the maximal dimension, right? So I would suppose maybe it's more than 5 cm. So at least it's a T3 and above. And at the same time, remember I said there's some the chest wall, there's some irregularities and not very clear demarcation. So probably there's some chest wall involvement, which is also part of T3. So at least a T3. Now we look at lymph nodes, right? So just now, remember, I specifically bring your attention to the supraclavicular lymph nodes. So when you have a supraclavicular lymph nodes, it will be N3. Okay, it'll be the N3, but unfortunately, I can't really comment much on the metastasis because we don't really have the full abdominal CT scan, only the lungs. But from the lungs, I don't really see any lesion in the contralateral lung. And yeah, so because once you have a contralateral lesion, you'll be M1A. Right, so here there's no contralateral lesion, and can't comment much on the abdominal organ. So from the whatever information that we have just now, we know that it's T3 and above, and also N3. So from this, we will derive that we can get this from this table that the patient had at least a stage 3C lung CA, right? At least 3C. So we will need to complete the staging and then to give a more accurate and proper staging. Okay, thank you. I'll pass the time back to the presenter.
Thank you, Dr. Dean, for your kind sharing. Okay, give me a moment. All right. Okay, so I'll continue on the procedures that had been done for this patient throughout the management of the case. So first off, the patient has actually done a thoracocentesis, whereby a picture catheter was inserted to relieve the pleural effusion, as we can see in all the imagings. So the pleural fluid cytology was done by using the cell block technique, and you can see here, this is the HPE results, whereby there were presence of blood and a few malignant cell clusters in the pleural fluid cytology. Next up, a patient has also done colonoscopy and also OGDS, indicated by raised CEA levels. On colonoscopy, there were actually no lesions seen, but on OGDS, there was actually erosive pangastritis saw. So this is the lymph node biopsy. So for the lymph node biopsy, it was actually done under local anesthesia, and the sample were taken from the supraclavicular lymph nodes. So it had shown metastatic moderately differentiated mucinous adenocarcinoma, and here are the HPE results of the case. Next up is the tumor immunoreceptor status that was done for this patient. So the thyroid transcription factor was negative, and there were no mutation of the epidermal growth factor receptor. So lastly, patient has also done bronchoscopy as his condition deteriorated with organ support in the ICU, and the diagnosis made was metastatic moderately differentiated mucinous adenocarcinoma. So that's all for the case presentation. So now I would like to invite Dr. Narasimhan to further discuss on the management as pertaining to this case.
Well, thanks for the detailed presentation. But I would be interested if any further scans performed to look at the abdomen.
As far as the information that we've got, I actually don't have the CT-TAP with me.
All right. Normally, if this patient were to present without a supraclavicular lymph node, and I'll be interested to know the adrenal glands, if it's one of the common sites where you get this lung cancer metastasize. And usually if you have the CEA elevated, and if you're dealing with lung cancers, then it shows that there's already a pleural disease. So even in pleural disease, you'll have CEA elevation, and you know that this could be already an advanced stage before even you do the surgery. So that is one of the things that we look for. This patient is obviously not a surgical candidate. He should have gone for chemo radiotherapy or even targeted treatment if we know the PD-L1 status. So I'm not sure if you have the PD-L1 status with you.
I don't think so.
All right. Okay. So he is not a candidate for any surgical intervention. Is there anything else you would like me to comment on this patient?
Yes, I do agree with all the points that you have stated, Mr. Nara. So actually, we would also like to ask a few questions pertaining to this case when we were preparing for the case, because as in the earlier session that we have listened to all the talks, actually EGFR and the mutation was highly associated with the pathogenesis of the lung cancer here. But also in this patient, there was a negative thyroid, the TTF1 status. So actually, do you think that there are any role of the thyroxine kinase inhibitor in this case?
Well, lung cancers primarily can present with TTF1 negative. Although TTF1 negative is an indication that it is a primary lung malignancy. So in this scenario, it will be very useful if you have got biopsy from the lung tumor itself rather than the supraclavicular node. Well, I guess it was not done in this patient because he deteriorated subsequently and needed ICU management. No doubt it's already an advanced stage, but it will be nice to know whether the biopsy from the lung tumor that we saw in the CT scan coincides with the supraclavicular lymph node, especially the TTF1 status. Whether TTF1 negative patients will respond well to TKI, I think Dr. Ivan can elaborate on that.
Okay. Usually for smoker, I think this patient is smoker, right? So usually they are not, they won't have mutation. But the chance of getting a mutation probably is of 10% and above. Sometimes we still can see some mutation in patient with a smoking status, smoker. But usually smoker is more, more appropriate treated with immunotherapy. But for this patient, I think the clinical scenario is a bit different. So I would like to know if during the presentation, what is the performance status of the patient? Performance status usually means, usually we score the patient for every cancer patient. So whether they are 0 to 4, so 0, they have no symptom at all. 1 is they have a little bit of symptom, but not affecting their, activity of daily living. But 2 is about, they can still work, but more than 50% of their waking hour, that means they are not sleeping, they still can work more than 50% of their waking hour. But 3 is they are a bit bedbound, but they still can walk. But more than 50% of the time they need to sit or lie down. 4 is bedbound. So this patient, they are in what category? Is a patient is bedbound, need to come in with wheelchair?
Dr. Ivan, I think this patient falls into category 1.
Category 1. Okay, but the patient after that rapidly deteriorate and end up in ICU.
Yes, but on presentation, he was category 1.
All right. Okay. So it's a bit different because performance data is very important. If this patient present with performance status of 3, then there's no active interventions recommended oncologically. But PS1, that's very different. But now this patient, I think the more important factor is the advanced age. So 80 something. So usually if you want to manage this patient, so you need to act very fast. So it means we need to send the investigation fast and get back the result fast so that can manage appropriately, because the tumor usually behaves quite aggressive. Especially my comment for this case, this is an atypical kind of lung cancer because it's TTF1 negative. So it's a typical mucinous adenocarcinoma, which is not very common, but we know that it is a more aggressive form of lung cancer. But clinically, I think it is lung cancer because look at the presentation and look at the scan. Usually we will scan the case with PET-CT. But we know that resource constraint, some of the case we only scan with a CT scan only. But this patient, I think, should be stage 4. I think it's a malignant pleural effusions. Pleural effusions with a malignant cell there, then it's stage 4 already. So in terms of the mutation testing, nowadays we usually will do a broad base of mutation analysis. Means it's the next generation sequencing. This patient only do the EGFR mutation. We usually will send for about 30 to 50 mutation testing, and then we get back the result within 5 to 10 a week. Then if there is mutation, then it make the scenes totally different. Then we can start a targeted therapy. Then we change the course of disease completely. But if this patient is PD-1 positive and is very high positive, like 50% and above, or even sometimes we get 80, 90%, then it will be the good candidate for immunotherapy. But for this patient, probably more than 80 years old, it's quite difficult to deliver chemo. So we can, if you can do all these tests fast, then if a patient suitable for immunotherapy, then will be a good candidate. Yeah. I think that's about it. And CEA, raised CEA is very common in lung cancer. And sometimes, actually a lot of patients with a raised CEA. So, and a lot of time you do a scope, you can't find anything. So for patient who have with obvious lung mass and everything, usually the IHC and HPE give you an answer rather than a scope. Yeah, I pass it back to the presenter.
Right. Thank you, Dr. Ivan. So just to add on, the patient was seen in a hospital in Myanmar. So they actually had to send the investigations to Thailand, and because of the pandemic, it was delayed to 2 weeks. It took 2 weeks to come back. Yeah. So the scoring actually deteriorated from 1 to 3 to 4 within 2 to 3 weeks.
Yeah, it's very common because lung cancer in Nigeria is progressed very fast. And actually some of the lung cancer, whatever you do, you are not fast enough. So sometimes the scenario is like that. Even you see the clinical trial with immunotherapy, you see a sharp drop of the survival initially, then it becomes stabilized. So some of the patients, they will deteriorate, whatever you do.
Okay, thank you, Dr. Ivan. Thank you, Dr. Narasimhan and Dr. Dean for your kind sharing. And I believe all of us have learned a lot. So we will now open the floor for questions.
Okay, yeah, so thank you very much for the great sharing. If there is no further questions, then maybe we can proceed to the photo taking section. Can I invite all the participants, including our panelists, to on the camera for us to take a photo?
Okay, so students and participants, if you would like to switch on your camera, I'll wait for one more sec. All right, so on my count, 3, 2, 1. Okay, one more time. On my count, 3, 2, 1. Okay, thank you very much, everyone.
All right, thank you.
So I think we have come to the end of our sessions for today. Hang on, sorry about that. Yeah, okay. So before we end, we would like to again invite Mr. Yan as the advisor for the entire webinar sessions to give his closing remarks. Mr. Yan, please.
Yeah. Hi, good afternoon, everyone. It has been a momentous day. Today we have discussed, shared, learned about the evidence-based research and innovation in the area of lung cancer by our experienced research scholar, Dr. Wang and Dr. No. It's been really thrilling, and this is our hope for the future emergence of new drug therapy for lung cancer. And again, we also experience and learn updated guideline in the management of lung cancer. Our specialists, inspirational clinicians, Mr. Narasimhan and Dr. Ivan, which is really comprehensive and really interesting. The Q&A session and case discussion by the specialists and speakers was really eye-opening and fruitful for all. May I express my appreciation to all the speakers, panelists for their valuable contribution to our CSCR webinar. And my deepest gratitude goes to all the participants who have to make it such a successful event today. My thanks are of course reserved for our two student champions, presenters, and volunteer for case presentation, and our panelists for case discussion for the priceless contribution. We successfully implemented a student centered case study session and webinar series. We will adopt this culture furthermore in the future events. Before ending my closing remark, I would like to convey my special thanks to CSCR Chairperson Prof. Alan and CSCR Chairwoman Dr. Ong, and our organizing committee members who have been extremely cooperative and staying with us beyond the time allocated. Finally, we really love to collaborate with the national and our international counterparts for our future webinars, cyber classroom, and CME program. We shall meet again in the future events. And thank you very much indeed for today's contribution. And stay safe and healthy. I would like to say goodbye for now. Thank you.
Thank you, Mr. Yan. So with that, we will end today's sessions. And once again, thank you all for participating and spending your half of the weekend Saturday with us, and have a remaining good weekend ahead. Thank you very much.
Thank you. Bye.
Bye.
Thank you, everyone.